Hello to all of you. We are going to discuss chapter number 9. So this is about glycolysis and fermentation. So I'm going to go over some of the important chapters which you already know, and it is not possible to make very big videos. I'm going to important, and this is a topic which you have been learning in classes also. So I'm going to skip all these things, what metabolic reaction pathways are, what catabolic pathways are, and what you know that. I would like to talk about here the structure of ATP, which is very important because basically cellular respiration is a process in which the ATP is extracted from the food we eat. And the energy which is extracted, it is extracted in the form of this chemical called ATP, adenosine triphosphate, is the energy in our cells, and you know its structure. So its structure is there is a ribose sugar. In ATP, the sugar is ribose. Then the nitrogenous base is adenine, and there are 3 phosphates. That's why it's called adenosine triphosphate. So phosphate, which is attached to the carbon number 5 on the ribose sugar, it's attached by the bond called phosphoester bond. But the bond between these groups, they are so they so keep them together. Bond really phosphates a lot of energy, and that is in the form of these bonds, phosphoanhydride bond. So this bond, which is between the carbon and carbon number 5 of the ribose and phosphate group, this bond is called bonds, and the other bonds, they are called phosphoanhydride bonds. So this is basically I wanted to know, let you know, and you know that when the ATP is hydrolyzed, it's converted into ADP, and in this process, a lot of energy is released. So this is a exothermic exergonic reaction. Okay, so I'm going to skip that part. So another important thing to understand in metabolism is oxidation and reduction reaction. So as you know that oxidation is the removal of electrons, and the reduction is the gain of the electrons. And you know that I actually have given you some summary about this chapter in our last lecture in our face-to-face format. If I remember, we finished the active transport, and we had some time, and I discussed the overall view of metabolism. So I already covered these things. So oxidation is loss of electrons, and you know that in the cells, the electrons, chemicals, they do not run by themselves. They are present in the form of hydrogen atoms, and the loss of electrons means the loss of hydrogen atoms, and this process called dehydrogenation. And the electron, so basically in this whole process, and the reduction is called hydrogenation because it's a gain of hydrogen atoms basically, and oxidation and reduction goes together. So one of the intermediate metabolite or one of the compound, it loses electron and the other one's gains. So oxidation and reduction process takes place together. And in this process of cell metabolism, there are some coenzymes which are basically are the electron carriers. They are the ones which actually take away the electrons, and they take the electrons to their final destination, which is the inner membrane of mitochondria. So these coenzymes, they get they get reduced, and then they deliver these electrons to the electron transport chain, which is present in the inner membrane of mitochondria. They deliver these electrons there, and then there they get oxidized. So these electron carriers, which is NAD plus and FAD, they basically carry the electrons from the different stages of the cellular respiration, and they deliver them to the inner membrane of mitochondria. So here is shown how this NAD plus carries the electron. So NAD plus, it has already one positive charge on it. So basically it accepts 2 electrons and 1 proton. So these are hydrogen atoms which are actually coming from the food. So these every hydrogen atom from 2 hydrogen atoms. So is used with NAD plus. And first of all, it's more hydrogen. So for to NADH, it has 2 electrons and it has 1 proton, and 1 proton is released. So this is the way these electron carry electrons. So next important thing I would like to talk about is basically we are talking about cellular respiration. So in this cellular respiration, so cellular respiration is an aerobic process. In this process, because oxygen is involved, and oxygen acts as the final electron acceptor. Oxygen acts as the final. And there is an anaerobic respiration also, and there is different cellular respiration, aerobic respiration. Cellular respiration or aerobic respiration is the same thing. Then we have aerobic respiration, then we have fermentation. So anaerobic respiration and fermentation both are anaerobic processes, which means that both of them takes place in the absence of oxygen. But anaerobic respiration and fermentation, these are two different processes. So anaerobic respiration is a process in which the final electron acceptor is not oxygen, but it is something else than the oxygen. It involves like iron or hydrogen. So different organisms, they can have different kinds of final electron acceptors. So like sulfur, protons, or ferric ions, they can be used as the electron acceptors. Fermentation is basically glycolysis, and so glycolysis is the first stage of cellular respiration, and it can take place in the absence of oxygen, and it takes place in the cytoplasm. So fermentation is basically glycolysis with one extra step. So fermentation is different from anaerobic respiration. So we are going to talk about the details of this. So let's first of all talk about the details of the glycolysis. So glycolysis, I would like to explain from this. So in the glycolysis, you need to understand all these 10 reactions. You need to understand what is going on in every reaction, and you also need to know the enzymes involved. So this is a list of enzymes which are involved in every reaction. So basically, this is the overall glycolysis. You know that in glycolysis, glucose gets converted into glucose 6-phosphate. So when the glucose is in the first reaction, when the glucose is getting converted into glucose 6-phosphate, here the cell has to actually spend the energy. So glucose, when it gets converted into phosphate, this phosphate group is given by ATP, and this takes place with the help of the enzyme called hexokinase. So here this is an investment phase because the cell is actually investing the energy here. Then glucose 6-phosphate gets converted to fructose 6-phosphate. So you know that glucose and fructose, these are basically isomers. So this conversion of glucose 6-phosphate to fructose 6-phosphate takes place with the help of the enzyme called phosphoglucoisomerase. And then this glucose 6-phosphate gets converted into fructose 1,6 bisphosphate. So there is a addition of one more phosphate group. Here also the cell has to spend some energy. So here also there is a investment of the energy of the cell, and ATP is involved. And this reaction takes place with the help of the enzyme called phosphofructokinase, because here is the the phosphate group is being attached here to this group, to phospho 6. So phosphate group is being attached to phospho 6 fructose 6-phosphate, and it is getting converted into fructose 1,6 bisphosphate. And then there is the involvement of another enzyme called aldolase, and cleavage of this compound takes place. This compound has 6 carbons. It gets cleaved into two compounds, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So it cleaves, this compound is cleaved into two compounds, and these two compounds, they have 3 carbons in them. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, both of them have 3 carbons in them, and they can get these are basically isomers of each other, and they can get converted into each other with the help of the enzyme triose phosphate isomerase. And actually in glycolysis, dihydroxyacetone is actually converted into glyceraldehyde 3-phosphate. So at this step there are 2 molecules of glyceraldehyde 3-phosphate. So after this, whatever happens, you have to multiply by 2, because dihydroxyacetone gets converted into glyceraldehyde 3-phosphate. So we have 2 molecules of glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. So glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate, and this, what is happening here is this compound glyceraldehyde 3-phosphate is actually getting oxidized, which means there is stripping of electrons, there is a removal of electrons, and those electrons, they are taken by NAD+, and NAD+ is getting converted into NADH. And here there is addition of phosphate, and this is taking place with the help of the enzyme called glyceraldehyde 3-phosphate dehydrogenase, because here dehydrogenation or oxidation is taking place. Then in the next step, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, And here what is happening is, you can see this is a compound with two phosphates. This is a compound with two phosphates. Now it's getting converted into a compound with one phosphate. Where this is going? This phosphate is actually going to ADP, and ADP is getting converted into ATP. Okay, and this is the first molecule of ATP which is produced. So multiply by 2, there are two molecules of ATP produced here, and this is, these two molecules of ATP, they are produced by the substrate level phosphorylation. So substrate level phosphorylation is the process in which ATP is produced from ADP. ATP is produced from ATP. ATP is produced from ADP, and it takes place with the help of the enzyme, and the enzyme is phosphoglycerokinase. And after this, the 3-phosphoglycerate is converted into 2-phosphoglycerate. So here is just the position of phosphate group is getting changed, and this takes place with the help of enzyme phosphoglyceromutase. And after that, this 2-phosphoglycerate is converted into another compound called phosphoenol pyruvate, and of enzyme enolase. And then phosphoenol pyruvate is converted finally into pyruvate. Okay, and here this is taking place with the help of enzyme called pyruvate kinase. And here what is happening is, the phosphate group is given to ADP, and this ADP is converted into ATP. So two molecules of ATP are produced here, two molecules of ATP are produced here, and these are in total 4 molecules of ATP are produced, and 2 molecules of ATP are used here. So the net gain of ATP is 2 molecules of ATP, and they're produced by the process of substrate level phosphorylation. And here you can see NAD plus is getting reduced into NADH and multiply by 2, because there are 2 molecules of glyceraldehyde 3-phosphate. So this is overall glycolysis you need to know. These are the important steps you need to know. You need to know the enzyme. You can have some questions on the enzymes. So this is glycolysis. Now so this is all what is explained is written here. You can go over by yourself. So now I'm going to talk about a little bit about fermentation. So there are 2 kinds of fermentations. So this is the path of aerobic respiration or cellular respiration we are going to talk about in the next chapter. So in the fermentation, basically there are 2 kinds of fermentations. One is lactic acid fermentation, and the other is alcohol fermentation or ethanol fermentation. So in the lactic acid fermentation, pyruvate is gets converted into lactate. So basically this lactic acid fermentation or lactate fermentation involves glycolysis. So from glucose, first of all, pyruvate is formed. So whatever is the yield of glycolysis is the same yield of fermentation. So in fermentation, it is a very inefficient way of producing energy, because in this process only 2 molecules of ATP are produced, because fermentation is basically glycolysis. From glucose, pyruvate is formed, and this pyruvate is actually getting itself reduced and it is forming lactate. So remember that in glycolysis there was one NADH. Those NADH, they need to be oxidized back into NAD plus. They need to be oxidized back into NAD plus, which means they need to be converted back into NAD plus, because otherwise the process of glycolysis will stop, because these are the electron carriers. So they need to be regenerated. So basically pyruvate gets reduced. It takes the electrons from NADH. NADH gets converted to NAD plus, and pyruvate itself gets converted into lactate. And on the other hand, in the alcoholic fermentation, pyruvate is converted into, first of all, into acetaldehyde. So pyruvate has 3 carbons. First of all, there is a release of carbon dioxide. This is 1 carbon. So pyruvate, which has 3 carbons, is converted into acetaldehyde. It has 2 carbons, and then this acetaldehyde is then converted into ethanol. So acetaldehyde is then converted into ethanol, and this basically this acetaldehyde is getting actually reduced and forming into ethanol, because it is getting electrons from NADH, and NADH is getting converted into NAD plus. So these are 2 kinds of fermentation, you know. Then we need to know the process called aerobic glycolysis. So this is also called Warburg effect. I have also posted one video for you, because this process is actually used by the cancer cells. This is called aerobic glycolysis. So this is a process in which basically the cancer cells, they undergo fermentation even they have oxygen available. So they prefer to do fermentation, they prefer to undergo fermentation, although oxygen is available to them. So why do cancer cells do that? Because fermentation is a very inefficient process of producing energy. Only two molecules of ATP are produced in this process. The yield of energy is very less. But why do cancer cells prefer to do aerobic glycolysis? Aerobic glycolysis is basically fermentation taking place in the presence of oxygen. They undergo this process because, as you know, cancer cells are very fast dividing cells. For the proliferation of the cells, they need a lot of carbon skeletons, and they undergo aerobic glycolysis mainly to produce carbon skeletons, because from these carbon skeletons they need to make more new substances which are going to be used for making new cells. Okay, this is some diagnostic thing. You can go over it. It's very easy to understand. So I will be now talking to you about um, like in our food, we not only eat glucose, we eat various things. So how do they give us energy? So whenever we talk about cellular respiration, we go and we basically talk about glucose, right? We are not only eating glucose in our food, we are eating various other things. So we eat various kinds of disaccharides, we eat various kinds of polysaccharides. So how do they enter into enter into this pathways and they yield us energy? Now, for example, this you can see the example of lactose. So lactose is a disaccharide made of glucose and galactose. So lactose gets converted into glucose and galactose, and glucose then after that, what happens to glucose. So what happens to galactose? So galactose first of all gets converted into galactose 1 phosphate, then it gets converted into UDP galactose, then UDP glucose, and then it gets converted to glucose 1 phosphate. And this glucose 1 phosphate so glucose 1 phosphate gets converted into glucose 6 phosphate, and then it goes into glycolysis, and after that everything is same. So sucrose gets converted into glucose and fructose. So fructose gets converted into fructose 6 phosphate, and it can enter into glycolysis. So similarly, glycogen, starch, these are all polysaccharides. They all first of all get converted into glucose 1 phosphate, and the glucose 1 phosphate can get converted to glucose 6 phosphate, and it can enter into glycolysis. All things which we eat in our food, fats or proteins, they first of all get converted into some intermediate metabolite, and that intermediate metabolites enter into glycolysis, and after that Krebs cycle, and then the oxidative phosphorylation. After that everything is same. So the last thing in this topic, I would like to talk about gluconeogenesis. This is the process how glucose is synthesized. So basically, gluconeogenesis is basically same as glycolysis. It is basically the reverse of glycolysis, but there are only few reactions which are different in this. Let me try to make it big. Okay. So all the other reactions, go back to the last slide. Okay, we are here. So three reactions which are different: this one, and this one. Other than that, all the other three, reactions are just the reverse of the glycolysis. So basically, how glucose is formed. So basically, it can be formed in the gluconeogenesis. It can be formed from pyruvate. So pyruvate first of all get converted into phosphoenol pyruvate. So this step is a little different in gluconeogenesis. First of all, pyruvate, which has 3 carbons, it's getting converted into oxaloacetate. So pyruvate has 3 carbons, oxaloacetate has 4 carbons. So there is an incorporation of carbon dioxide, so which means there is an incorporation of 1 carbon, and this compound gets converted into a compound with 4 carbons. So pyruvate has 3 carbons, oxaloacetate has 4 carbons, so there is an incorporation of 1 carbon dioxide, and this step needs energy. ATP is converted into ADP. This involves the enzyme called pyruvate carboxylase, and then this oxaloacetate is then converted into phosphoenol pyruvate. There is again loss of carbon dioxide, and there is again input of energy here, and this takes place with the help of enzyme phosphoenol pyruvate carboxykinase. Then other than that, all these steps are just the reverse of glycolysis. Then here is another step here in which fructose 1,6 phosphate is converted into fructose 6 phosphate. It involves an enzyme called phospho 1,6 bisphosphatase. And in this, fructose 1,6 bisphosphate is getting converted into fructose 6 phosphate, which means it is losing phosphate here. And this is taking place with the help of the enzyme phosphatase. So this is a little different from glycolysis, and then this step is also different. So in this step, glucose 6 phosphate is getting converted into glucose, and this is taking place with the help of enzyme glucose 6 phosphatase. So here again there is a loss of phosphate. So this is basically gluconeogenesis. So that's all in this chapter. So if you don't understand anything, again you can send me email, or you can also have a virtual meeting, but you have to let me know 1 or 2 days before so that I can fix the meeting. Okay, I've set up the meeting there. Okay.